Welcome to BioVivacious. BioVivacious is a YouTube channel dedicated to clear fundamental concepts in biosciences. Today we are going to discuss about the Calvin-Benson cycle. We are aware that the electron transport that is taking place in the oxygenic photosynthetic organisms produce two products. And the two products are ATP and NADPH. So these are energy-rich intermediates. But these intermediates, they are transient, they cannot be stored for a long time, or they cannot be used for generating a biomass. Therefore, plants must look for something which is stable, which can be stored as starch or sugar or as protein or oil in certain plant species. So they must look into converting the energy into a more stable form. It is in this context we are trying to understand how carbon is going to be fixed and that can serve as sugar or a starch for generating biomass for plants. This is a pathway that was developed somewhere in the 1940s to 1950s by brilliant experiments carried out by several scientists, and two important scientists who contributed to the understanding of this reductive pentose phosphate pathway is the Calvin and Benson. Calvin says in his autobiography that this was a chance discovery. He was waiting for his wife in the car while she went for shopping. So this idea struck him that this is how probably carbon fixation in plants occurring. Then he carried out several experiments. We are going to look at those experiments which made a significant contribution to our understanding of the Calvin-Benson cycle. So this experiment was popularly called the Lollipop experiment. Lollipop experiment. Let me draw the figure of this lollipop and then I'll explain to you how this experiment was carried out. Calvin and Benson used this apparatus. This was specially designed in order to carry out the fixation of carbon dioxide. So they used two techniques. The first technique which they used is two-dimensional paper chromatography. They also used another technique that was autoradiography. You should understand that this is a time immediately after the Second World War. A lot of radioactive materials were available for research. So they used these radioactive materials in order to understand fixation of carbon dioxide. So they designed this experiment, this looking like a lollipop, so therefore it was known as the lollipop experiment. So this is a glass chamber, and in this glass chamber they filled with a chlorella, that is a photosynthetic algae. So this is filled inside, then it has an outlet for air and an inlet. They are able to inject carbon dioxide, which is C14 labeled, radio labeled carbon dioxide ion solution. They are able to pump inside, and they will illumine for a few seconds. When illumine, the photosynthesis, carbon fixation will occur. When it is illumined, the photosynthesis will occur. Then what they will do is, at definite times, maybe after 2 seconds, 5 seconds, maybe after 60 seconds, they will collect in the sample in order to understand how much of carbon dioxide has been fixed. is the radio labeled carbon has been incorporated within the structure. So in order to do that, they will collect a sample into hot alcohol. When chlorella is transferred into hot alcohol, what happens is all metabolic reactions will come to an end. This sample is then analyzed by using two-dimensional paper chromatography. Let us look at how the paper chromatography has been carried out in order to prove how carbon dioxide is fixed. This is just an example of a two-dimensional paper chromatography that was carried out. Now, for example, this is the origin where the sample was loaded, that is the chlorella, which is collected into hot alcohol, is loaded here. This is the one dimension where the solvent system used is butanol, propionic acid and water. So therefore several components will get separated. Then it is run on another dimension, that is phenol, water, another dimension it is run. So therefore the components got separated. Initially they carried out for 2 seconds if the light was shown, and then this paper chromatography was carried out. They found many compounds were separated. Then they carried out, increased the duration of exposure to light, maybe to about 60 seconds. Then they found many more compounds are getting separated, and they tried to identify these compounds. They realized one of the compound which is formed is 3-phosphoglycerate, a 3-carbon compound. So the assumption was, in order to get a 3-carbon compound, there must be a 2-carbon compound to which a CO2 molecule is added. So therefore they will be able to get a 3-carbon compound. They began looking for this 2-carbon compound, but they could not find a 2-carbon compound. That led to the assumption that there must be a 5-carbon compound to which a 1 carbon CO2 is added, that will give rise to a 6-carbon compound which may be unstable, that is split into 3-carbon compounds. This is how they began looking for a 5-carbon compound and came across a molecule known as ribulose 1,5-bisphosphate. Today we know that it is to ribulose 1,5-bisphosphate, carbon dioxide is attached, which will split into 2 3-carbon compound, that is nothing but 3-phosphoglycerate. This is in the fixation carboxylation reaction. Now they elucidated the entire pathway. For all the work which they carried out, they were rewarded Nobel Prize in 1961. Kelvin was awarded Nobel Prize in 1961. In 1989, President George Bush acknowledged his contribution by giving him the National Medal of Science. Kelvin also wrote his autobiography, Following in the Trail of Light, in 1992. What we have seen now is the experiments and the pain they have undertaken in order to understand the fixation of carbon dioxide and for the production of a starch. We will be looking elaborately at how exactly these reactions are carried out.